It's time for an anatomy lesson. Well, more specifically, the anatomy of a microscope. We're going to look at the different parts of a microscope and better understand how we can use this as a tool to see an invisible universe that's all around us. Now, when we think of microscopes, there's all kinds of them. And if you haven't bought a microscope yet, what kind of things should you consider that's going to help you to purchase the one for your specific needs? Well, first of all, let's take a look at this microscope right here. We're going to look at this part because this is the part we look through. See, I've got one right here. Notice this one is too. This is called a binocular microscope. And this portion, specifically the part that you put your eyeballs up to, that is known as the eyepiece. So here we got two eyepieces, and some people prefer this kind of a microscope, especially if you do a lot of lab work, because it's going to help reduce eye strain. You'll notice that some of these microscopes actually have a rotating disc up here. If I'm looking at this and I want to show a student, or a student wants to show me something, you don't actually have to move the microscope at all. It sits stationary. When you move it, sometimes it jars and you miss where the specimen is. Things move away from it. You simply just rotate that. This is called the eyepiece, and that's going to be a very, very important thing to keep clean, protect, because that's going to be part of your visual experience. Now, the eyepiece itself has a lens to it. One thing that we like about microscopes is they magnify itsy bitsy teeny tiny things so that we're able to see them up close and personal. If we look at this eyepiece, as a general rule, they have a 10x magnification. In other words, whatever your image is that you're looking at on the slide, it's going to increase its size 10 times. Now you may say, well, that's more than enough, right? I don't need to have it bigger than that. But keep in mind, when we're thinking of the prefix micro from microscope, it goes back to the metric system and we think of 1/1,000,000th of something. In this case, it's 1/1,000,000th of a meter. A meter is about the size of a man's arm, and if you cut my arm up into 1,000,000 equal sections, don't try that at home, that would really hurt, by the way. We're talking about little teeny tiny things. In fact, when we think of red blood cells you can't see with the naked eye. You can see the blood as a fluid, grossly, that type of thing, but you can't see the individual cells because they're like 6 microns, 6/1,000,000th of a meter. Yowzer, that's really, really small. So we've got this first part we want to pay attention to, and this is known as the eyepiece. Now, the next thing is known as the body tube. The body tube is actually the cylinder that connects the eyepiece to the microscope. And wherever you keep your microscope, you may want to put some type of a piece of plastic or something over it so you keep the dust off of it. And anything that's going to cause the lenses to become cloudy or difficult to see through, your specimen is not going to have the same detail and sharpness that you really want it to. Now, let's take a look at another part. This part right here is connecting the top portion with the rest of it. This is known as the arm of the microscope. And the arm is going to be very, very important, not only for support, to hold everything together, but when you're picking up your microscope, these things are delicate instruments. You want to pick it up by the arm and support it by the base. And it's a good idea to hold it close to your body as you're carrying it. What you don't want to do is swing it back and forth, bang this, bang that. Next thing you know, things are going to be out of kilter and you're not going to be able to see very well. So you've got the arm here, and obviously where it's sitting, that's known as the base. As we continue downward, let's take a look at the wheels on these things. Here's a big wheel, here's a small wheel. Notice on this microscope, both of these wheels are part of the same mechanism. There's a little one there, there's a big one there. Well, what's the big wheel versus the small wheel? This is going to be important. Well, if we look at the big wheel, this is called the coarse focus. Coarse focus. Now, when do we use that? We use the coarse focus only for using low magnification. See, when we think of a microscope, we have multiple lenses, so we have multiple powers to our magnification. When we're looking at the lowest magnification, and that would be if you look at these little cylinders, we'll talk about these in just a moment. You see, it's really small. That's a low power. When we go to the intermediate one, that's a power greater than the small one. And then this one, we have 3 of them. This is the biggest power. You only use this coarse focus, this wheel right here, when you have the lowest magnification closest to the stage, the platform that you're putting the slide onto. Notice that as you move this thing, it's going to cause this platform right here to move up pretty radically. I mean, it's easy to see that this is moving up and down. And as you're moving it up and down and looking through your eyepiece, that's going to help you to get things into focus or out of focus. So we'll get some practice as we're going through this over the next several labs. The little wheel, on the other hand, is not called the coarse focus. It's got another name. It's known as the fine focus. The fine focus is going to help you to see the image much more detailed, sharper, at higher magnifications. So now we're looking at the little wheel, and this one we're going to see moves the slides up and down just like we talked about before, but as I'm turning this, you probably can't see much movement at all. It's going to be much less dramatic in terms of going up, going down. Now the question is, why do you have to make a distinction between high magnification and low magnification? Think for just a moment about putting your slide on this platform right here called the stage. You got these little clips, talk about that in just a moment, and you lay your glass slide on here. If I go ahead and I start with the low power, and I go ahead and use this, I can easily get this thing into focus, but notice I cannot run the stage into this. This thing stops before we ever even get close to this little objective or low power. If, however, I'm at a higher magnification like right here, or let's say we even go greater like that, now when I go ahead and I go up, notice I can bump right into the stage. Now what's that going to do? Well, that can either break my slide, and that's a bummer, right? Because that's the thing I'm using to see my little organism or tissue or cells or whatever, some structure. But we could also break the lens that are found on these individual powers. Don't want to do that. That's an expensive mistake. So anytime we're using high power, so these big tubes versus the little dinky one that I talked about earlier, we want to make sure we're using the fine focus. So don't forget that. I've already mentioned that the portion that the microscope sits on, that's known as the base. Nothing too tricksy there. Now, let's take a look at the interesting part. This is the part that rotates. We already talked about some eyepieces rotate, just like that, but we also have this rotation. Kind of interesting. This is known as the revolving nose piece. Man, all these funny names. Revolving nose piece. And it revolves because it's going to allow you to adjust the magnification based on the specimen that you're looking at. So we always start with the lowest magnification. That's going to be the smallest cylinder that we're talking about there. We need to make sure that as we rotate it so it clicks in position. I can just feel it. I don't even have to hear it click, but I can tell just by the feel that just clicked in place, and I can hear it also. If you try to look through the eyepiece at some slide and it's not clicked into place, it's off just a little bit, you won't see anything. It'll look very, very dark, even though you have the light source on. So you always want to make sure you have it clicked into place. I have students every year, they can't find anything. And the reason is their objective, the tube, is not lined up with the light source coming up from below. So what is the name for these little tubes that we're talking about that have different magnifications? Well, they are called objectives. Objectives. So the objective of your microscope is to find your specimen with the right magnification. So let's see how this all works, right? We have three different objectives on this microscope. We have the smallest one, that's the lowest power. We have an intermediate size, and then we have the biggest. Some microscopes that you can purchase will actually have 4 objectives. And so there's even a greater magnification typically you can find here. Each one of these objectives contains a lens with different powers. So the lowest one we use with the coarse adjustment. The higher powers we're going to use with the fine focus. Now, how do we figure out what the magnification actually is? If you take a look at these little cylinders, these objectives, they have numbers written on them. And if you look at the very smallest one, most likely you'll see something like 4x. Now, I've seen enough microscopes over the years to know that they don't always use this standard, but many times they will. So look at the number, let's say it's 4x. We've already talked about this one up here in the eyepiece is 10x. Well, how do you figure out what the total magnification is? You take the 10x and multiply it times the objective. So in this case, if this is 4x, it's 4 times 10, that would be 40 power. Many times the intermediate objective, which would be this one right here, is 10x. So this is 10 times 10 gives you 100 power. If we go to the third one, this is 40x. This is still 10x. The lens of the eyepiece is always 10x. So 10 times 40 gives you 400 power. That's pretty easy. So just multiply the lens of the eyepiece times the lens of the objective that you're using. That gives you your total magnification. If you have a microscope that has the fourth objective, many times that will be 100x. So do the math. What's 100 times 10? That's 1,000 power. Now, if you have that, we call those an oil immersion objective. And you actually have to use a special little type of oil over the top of a slide for you to be able to focus with that lens. Typically in our biology class, we don't even fiddle with that because it tends to be kind of messy, and one of the problems students have is cleaning the lens off after they've used the oil. See, if you go ahead and you bump this lens into the slide with oil in it, the oil actually gets onto the lens itself. And so you have to use a special kind of paper known as lens paper to clean it off. You never want to clean off any objective that you maybe got something on with Kleenex or paper towels. It's too coarse, can scratch it, and guess what? All of a sudden now the image you're looking at is going to be blurry versus having a very sharp focus. Moving on. Let's take a look at this platform. I already mentioned it does have a special name, and it's called the stage. So it's the stage that we're going to be viewing. All the world is a stage, right? Well, it is if you're a microbe. So here we've got this stage, and we're going to have a slide that we're going to place right on the surface. One of the things to keep in mind if you're looking to purchase a microscope is, number one, you want to get one that has at least 400 power, one that has a 40x objective, times the 10 is 400. That's a pretty decent powered microscope. In addition to that, you'll want to have something that you can kind of put the slide onto that makes it easy to follow things. If you look at this microscope right here, this actually has clips. And these clips, the way they're designed, you simply lift the clip and then you're going to slide the slide underneath it and it holds it into place. Now that certainly is adequate for most slide observation. But the reality is, one of the things we do in our class, which is a lot of fun, is we look at pond water where you have living organisms moving around. And these things like to swim. And so you have to be able not only to find them and identify them as something that's really kind of tough to do because there's such diversity that's associated with them, but if they're swimming, you got to be able to trail them. You got to know which direction you go. If you simply have these kind of clips like this, then you got to be pushing with your finger as the thing is moving along. And it's easy to lose them. There are some microscopes that have little wheels on it, and the wheels actually is what's moving the slides either from side to side or forward and back. You know, if you want to get a better microscope, get the kind with the little wheels that actually allows the slide to be positioned that way versus with your fingers. So we've got the stage, and that's going to be very, very helpful holding the slide in place. Underneath the stage itself, you're going to find out that there's a wheel there. And this one, you can kind of see, I've got this wheel, I'm kind of moving it back and forth. The wheel, as you turn this thing, is going to adjust the amount of light that's actually coming up from the bottom, from the light source itself. This wheel is called the diaphragm. And the diaphragm kind of works like the pupil in your eye. If you're in a very bright environment, the pupil gets really small. It restricts the amount of light that goes to the back of your eye where your retina is. On the other hand, if you're in a dimly lit environment, then all of a sudden the pupil expands, it dilates, letting in more light. In the same way, you're going to find that as you're looking at certain specimens, the light is too bright. And all you really need to do is rotate it so the little circle underneath restricts the light. If on the other hand, your slide seems to be kind of dark, two things to consider. Number one, have I turned my microscope on? Every once in a while I have a student who's embarrassed after I tell them you got to turn the light on first. But the other thing is if the light's on and it's too bright for that actual specimen, you can actually dim it. So that's what the diaphragm's all about, regulating the amount of light that passes through it. So take a look at it, and it's this little dial that's sitting right underneath here. Just kind of goes, and you can feel it click into place also, just like we saw with the revolving nose piece. So far, so good. There's another structure I want to mention that sometimes is present for some microscopes and some others, they don't have it. This one doesn't have it, unfortunately. It's called the condenser. And the condenser actually has this unit underneath that moves up and down. And you can see over here, we're pointing to it, so it's down underneath here. It's not the wheel that's sitting right in this region, but it's actually a whole unit right there. And what this is going to do is it's going to help to bend the light, and it helps with contrast. Helps you to be able to see the light that's passing through the specimen, and it gives you just another option so that you have clarity, sharpness, the ability to really see what you're looking at. You can kind of look at this, and it's one of those bells and whistles. Some microscopes have, other ones don't. All right, now, light source. How important is that? Well, it's like really important. You want to make sure you have a microscope that you purchase that has a built-in light source. Whether it's battery operated, doesn't matter, except you got to change the batteries, or you plug it in. I like the plug-ins because you don't have to fiddle. But you want to make sure you have that versus the kind where you've got like a little reflector or mirror underneath it, and you got to position this thing so it catches the light. Makes it a little bit more of a hassle.